Hi, my name is Poirier. I'm a bioscience technology student at Holland College for the year 2021. My research project is looking at the analysis of Kefir-Derived bacteria as an antipathogenic treatment, all determined through quantitative PCR. So for starters, kefir is a fermented milk drink, usually produced through the inoculation of milk with colonies of bacteria or yeast found within a kefir grain. Following this inoculation, it's incubated at ambient temperature for about 24 hours. The bacteria is then strained from the milk, producing a bacterially rich drink in the process, like this. This drink has become a worldwide phenomenon, mostly due to the health benefits thought to result from its ingestion. This includes microbiota modulation, antipathogenic effects, and even cancer prevention as well. Despite its popularity, though, many of these benefits haven't really been confirmed by experimental processes, remaining upheld by public belief or even tradition. Extensive research has been conducted with kefir, but many of its results remained inconclusive or couldn't be applied to the majority of unique kefir batches. To conclusively determine the true health benefits and microbiota modulation from kefir and its bacteria, a new method has to be used to quantify each of these aspects. My research project focuses on the quantification of kefir's antipathogenic properties through a new method using quantitative PCR on bacterial cold cultures. I hypothesized that Streptococcus mutans, a pathogen, would exhibit inhibited growth in co-culture with a kefir-derived bacteria. All of this could be quantified through qPCR. My methodology included the growth of bacteria from kefir and its isolation on skim milk agar. I also did some gram staining as well to help me identify my bacteria, and following 16S rRNA amplification through PCR and DNA extraction, I also was able to complete some genomic sequencing on it as well in order to help me with the identification. Following this, I grew my bacteria in co-cultures, partially for the comparison of biomass and observable inhibition, but really as a means to an end in order to do DNA extraction and qPCR in order to determine the amount of bacterial inhibition for both S. mutans and B. longum. I also did a zone of inhibition trial as sort of another outcome measure to show me any sort of bacterial inhibition that kefir isolated bacteria may have. Looking at my results here, I ended up getting a gram-positive coccus bacteria showing up in my gram stain, which is later corroborated with my genomic DNA sequencing, which told me I had Enterococcus lactis. My qPCR results didn't really turn out as I expected. My primers didn't give me any specific amplification. It was a real shame too, considering this was my main outcome measure. I did, though, look at two other outcome measures as well here. First off, I had my bacterial co-culture and observable inhibition. This was looking at the biomasses at the bottom of each of my co-culture tubes. A single blind participant ranked these, and I used a Kruskal-Wallis test in order to compare these non-parametric data values. I also did zone of inhibition as well, measuring the amount of area where B. longum and S. mutans didn't grow compared to a positive control of ampicillin. Both of these results gave me negative results where I didn't really see any sort of inhibition on S. mutans and B. longum from a kefir-derived bacteria treated on, which is a shame, especially when you compare it to a positive control such as ampicillin. In conclusion, my main outcome measure through quantitative PCR to compare bacterial inhibition on these co-cultures couldn't really be completed due to some procedural difficulties, despite loads of troubleshooting. My additional outcome measures resulted in no inhibition of either bacteria following treatment with my kefir isolate. Though these measures may be inaccurate and inefficient, really, my research work concludes that the kefir isolate Enterococcus lactis, which I identified, didn't significantly inhibit either bacterial growth. But it really illustrates the importance of co-culture and qPCR for the real accurate analysis of kefir microbiota modulation. So, moving forward, optimizations of my DNA extraction process and quantitative PCR must be made for additional attempts to accurately quantify bacterial inhibition through the treatment of kefir-derived bacteria.